Good afternoon, everyone. Today in our presentation, we'll talk about cloning. My name is Charlotte Deby, and I'll introduce the subject. Cloning is a natural or artificial process to produce genetically identical copies of living things. The copied material, which has the same genetic makeup as the original, is referred to as a clone. Researchers have cloned a wide range of biological materials, including genes, cells, tissues, and even whole organisms, such as a sheep. Sometimes, cloning happens naturally in nature, when a cell replicates itself asexually without any genetic alteration or recombination, for example. It's also the case of prokaryote organisms, which means organisms which don't contain cell nucleus, such as bacteria. Indeed, they create exact copies of themselves each time they replicate themselves. In humans, identical twins can be compared to clones as they shared almost the same genome. These twins are produced when a fertilized egg splits, creating two or more embryos that carry almost identical DNA. Identical twins of nearly the same genetic makeup as each other. However, they are genetically different from either parent. Nowadays, scientists also make clones in labs. They often clone genes in order to study and better understand them, but they can also clone animals to get gene mutation that help scientists to study diseases that develop in the animals. It's important to know that there are three different types of cloning. Gene cloning, which produces copies of gene or segments of DNA. The reproductive cloning, that reproduces copies of whole animals. And the last one, therapeutic cloning, which creates embryonic stem cells for experiments aimed at creating tissue in order to replace injured or diseased tissues. It's also very important to know that human reproductive cloning is universally condemned because of the psychological, social, and physiological risks associated with cloning. Now, Louise will tell us more about the history of cloning and also cloning in fiction. Then Sarah will tell us how it works, and she will also talk about the ethical issues related to cloning.
Hello, my name is Louise and I'm going to talk about a little history of cloning. So we cannot talk about cloning without mentioning Oswald Harvey, who discovered that the genetic information of a cell was carried by DNA in 1944, and the discovery of the structure of DNA in 1953 by Crick and Watson. But it was in 1903 that the term cloning was used for the first time, originally referring to plants reproduced by asexual reproduction. Moreover, in 1952, the first cloning of frog embryonic cells was carried out, some of them even developing into tadpoles. 10 years later, in 1963, the first artificial clone resulting from this technique is a carp. Then, in 1986, the first cloning of mammals from embryonic cells takes place. In 1996, Scottish scientists cloned the first animal, a sheep they called Dolly. She was cloned using an udder cell taken from an adult sheep. Since then, scientists have cloned cows, cats, deer, horses, and rabbits, but they still haven't cloned a human. This is partly because it's difficult to produce a viable clone. With each attempt, there may be genetic errors that prevent the clone from surviving. It took scientists 276 attempts to successfully clone Dolly. Unfortunately, she died in 2003 from a lung disease that sheep normally have at the age of 11 or 12. Other mammalian species quickly followed. In 2000, the first cloning of monkeys using the same technique that resulted in identical twins. The first cat was cloned in 2001. Similarly, in 2001, the first transgenic pigs were cloned. This experiment could be at the origin of the development of living organ factories aiming at being transplanted. Rabbits were cloned in 2002, and the first small mammal, a mule, was cloned in 2003. In 2004, a bull was cloned from a previously cloned bull. This can be called serial cloning. The first cloned human embryos were produced in 2001 when a private company, Advanced Cell Technology, produced 6-cell embryos. There are of course other important dates for cloning. However, if I mention them all, the presentation would be too long. And now I will talk about cloning in fiction. We can say that cloning has been and it still is widely used in fiction. Indeed, it is present in many films, series, cartoons, books, but also comics and video games. Truly, in the fiction world of Star Wars, clone soldiers are soldiers cloned from the DNA of Jango Fett, one of the most famous bounty hunters in the galaxy. His DNA has been modified to take the clones more obedient and subject to military discipline. Moreover, in the Orphan Black series, broadcast in France since 2013, the story revolves around Sarah, a young woman who, to escape her problems, gets into the skin of a woman who looks strangely like her and was put an end to her life in front of her. Moreover, in literature, there is often talk of cloning humans to save the world. Also, in the Superman universe, Lex Luthor tries to clone Superman, creating Bizarro, a failed copy of Superman. And finally, cloning is discussed in the Hitman video game.
I'm Sarah Couvrand and I speak about how does it work. So we have two types of cloning, the reproductive cloning and the non-reproductive cloning or therapeutic cloning. First, the reproductive cloning means reproduction of identical organism in order to create a clone out of natural procreation. The technique which can be applied to humans is nuclear transfer cloning. It's the same types of cloning used to create Dolly. The nucleus of a somatic cell of the first organism is transferred in the oocyte without genetic material. This egg cell is cultivated in cellular culture and it grows to the blastula stage. If all things are good, the clone has the same material genetic material of the cell donor. Next, the reproductive cloning or therapeutic cloning starts by the creation of the human embryo from the oocyte and the diseased cell. The embryo will grow to the blastula stage. It's a totipotent cell. It's about to grow to 300 cell types. This affection can be treated by transplants, by cell transplants. ethical issues related to cloning. Ever since the advent of cloning, there have been arguments for and against this process. Indeed, this process leads to numerous questions, particularly about moral ethics, as reproductive cloning presents the potential of creating a human who is genetically identical to another. This can conflict with religious beliefs and social ethics about human dignity, individual freedom, identity, and autonomy. Therapeutic cloning would require the destruction of human embryo in the test tube. Consequently, opponents argue that this technique to collect embryonic stem cells is wrong, regardless of whether such cells are used to benefit sick or injured people. So the reproductive cloning remains condemned, primarily for the psychological, the social, and the physiological risk associated with cloning.
To conclude, we can say that cloning is recent and that humans are afraid of it. The discovery of the genetic functioning of organisms is relatively new, and the expansion of this field is in full swing. Moreover, for several years now, cloning has been a subject much used in fiction, whether in the cinema or in literature. Finally, cloning remains at the heart of many controversies, in particular in the ethical issues of this subject.